we can start our third research briefing session. Third and last. Okay, wow. So this session is going to feature 4 members elected last year in 2022. And we've already heard in the previous session speakers from Class 1 and Class 2. Here we're going to hear the speakers from classes 3 through 6. The first speaker, Michael Shelley, is the Director, Center for Computational Biology, Flatiron Institute, Lyttle Professor of Applied Math, Courant Institute, and at NYU. He represents Class 3 section on Applied Mathematical Sciences, and his title is Active Transport and Self-Organization in Living Cells. Dr. Shelley.
Hey, Dr. Wessler. I can tell I'm mic'd up. So I'm very honored to have been asked by my section to give this talk, and I've really been having a good time at this meeting, meeting all of you and just kind of catching people in the receptions and finding out what they do. So thank you all. So what I hope to communicate to you today is how applied mathematics, mathematical modeling, simulation analysis can maybe shed light on interesting problems in science. And in this particular case, I'm going to be talking about some biology problems, which have some overlap with Julie Theriault's talk earlier. I joked to her earlier as I was going to say my first thing, I am not Julie Theriault. But there it is. So we were asked to give something about our path that led us into a life in science. And for me, you know, this is a path that you traverse in only one direction, so I decided to call it a trajectory. And the first thing I want to say is one of the the fortuity of it was taking calculus in high school from Mrs. McDonald, who was a very tough, vibrant teacher. She threw erasers and chalk and all these kinds of things. But she really instilled in me a love of mathematics, which carried me on to other places after that. And so you can see where those were. And one thing about calculus that really stuck with me was the ability that you could actually calculate things. And I think that in what I do, you know, like to calculate the volume of a vase, you know, you're 16 and you can calculate the volume of a vase without filling it with water and pouring it out and all these things. So, but I think that led me, wrong way. That led me naturally into computational fluid dynamics, and that's been one of my mainstays. And I'm going to tell you about problems today that really have to do with fluid flows and fluid structure interactions as they happen in cells that are developing, embryos and oocytes. But more along that path, I would say that one of the other big things to happen to me after Mrs. McDonald was that I, with Steve Childress and Jun Zong, founded the Applied Math Lab at the Courant Institute, which is a rare experimental lab in a mathematics department. It's not unique, but rare. And there we've had a 25-year experiment in combining, experiments with simulations and theory to look at, I think, a whole host of what I found to be very interesting problems that, again, carried me further, like questions of why do flags flap, how do flexible bodies streamline themselves, and especially active bodies in flows, swimming and flying problems, hovering problems. And I became more and more interested in the way in which moving bodies and flows interact with each other. And so I started to think about problems in what you would call active fluids and elements of the cells and cytoskeletal mechanics and things like that kind of naturally took me along. And so I'll tell you about a couple of transport problems, really, kind of self-organized transport problems. On the left, that first movie in green, what you're seeing is the positioning of nuclear material inside of a nematode embryo. I'll tell you about that. And on the right has to do with another type of transport problem where the fluid itself is transporting stuff around, nutrients and development factors that an egg cell needs to kind of move on with its life. Okay, so as I said, I started to think about these types of problems, and I think there we go. There were two sets of experiments thinking about how bodies interact in fluids that really gripped me, and I just want to mention them very briefly. And one has to do, well, what if you have hundreds of thousands of swimmers interacting with each other? And there was a beautiful set of experiments coming from John Kessler and Ray Goldstein, and on the types of coherent structures that you see when you put many, many bacteria in a droplet and you let them interact. And the way in which they're interacting is that they form, or rather the consequence of that, what they form is for them, large-scale vortices and jets and all these kinds of things, for which you would see transport speeds that were, say, an order of magnitude faster than any individual bacterium. So it's the fluid that's kind of organizing these structures. So this is kind of elements of self-organization. And so these bacteria are little swimmers. Because of the fact that you have no inertial forces being important in this problem at all, they turn out to be force dipoles, which means that we're dealing with the low Reynolds number world where inertia doesn't come into this at all, and it has its own peculiarities. Another, about just a couple of years later, was something that seemed very different but turned out to show many of the same types of effects. And this was coming from Zonomir Dojic's lab, then at Brandeis, where they took purified systems made of elements that come out of the cell, little stiff pieces of polymer called microtubules, threw them together with evolved motor proteins that would couple them together and exert forces. And these would self-organize into bundles, and these bundles would interact, they'd split, they'd merge, they'd produce large-scale fluid flows that in many ways was actually very similar to the other. So I started to think about these problems. So at the time, I had a very gifted postdoc, then postdoc, now I think lifetime collaborator at this point, David Santillan, who was working with me at Courant, and we decided to put together a numerical model of a bunch of swimming bacteria. So here we had a very simple kind of physical conception. We had a drag element, we had a thrust element, it produces a flow around it. We took 10,000 of them and threw them into a numerical box, a digital box. We let them do whatever they wanted to do. And it turned out that what they did was to organize into structures that were very similar to those that you actually saw in the experiments. So that was interesting, I thought. Local alignment, little packs of bacteria, running around fighting with each other, little gangs. And so you can't stop there, right? So, you ask questions about the stability, transitions of states, and these kinds of things. And so what we did next, David and I, is we confronted as mathematicians, we confronted the micro-macro problem. We constructed a theory, a kinetic theory, it might remind you of Boltzmann, where we have microscopic dynamics that we embed into a Fokker-Planck equation for the evolution of a distribution of positions and orientations, and we let that produce a stress in the fluid that drives the flow through. For those of you who are familiar with the Navier-Stokes equations, you'll notice there's no inertia. This is just the bare Stokes equation for viscously dominated flow. And out of that, it turned out that you could get incredibly complicated dynamics that, again, was very reminiscent of what you would see in experiments and in our previous theory. More than that, they gave us a route to do lots of kind of mathematics of analysis, transitions of states, loss of stability, kind of nonlinear stability problems. These days, it occupies a small group of PDE analysts who like to look at these, so that's nice. So now, I talked about that problem of the synthetic suspension of microtubules and motors and these kinds of things. Where did that come from, and why would you think about doing that? So what you're seeing here is a very important self-organized structure within cells. It's called the spindle. And the spindle is a self-organized football, if you like. This one's about 50 microns in length. It's a rather small football. But it self-organizes itself, and Julie referred to cell extracts. This is a self-assembled spindle because you threw a little plastic bead coated with DNA into it, and bam, out of this extract grew a spindle. And this will persist for hours and hours. And so these are the elements that actually are sitting inside of these synthetic suspensions that seem quite turbulent. Well, this seems pretty well behaved. It's very interesting that it lives for hours. But one difference between the two of them is that in the cell, microtubules, these structural elements, are very transitory. They live for about 20 seconds. But they are the substrate for exerting forces and organization and things like that for an object that goes hours and hours and hours. See, I'm kind of naturally migrating towards my non-existent laptop so I can move to the next slide. So we are studying the elements of the spindle quite a lot. It's one of our preoccupations. It's very technical. I won't talk about the mathematics of that, but I will tell you that what this has provoked us to do is to construct new classes of numerical methods. I come from a CFD background that let us look at how elements of the spindle realized digitally interact with each other by motors, model motors, that are making them rub up against each other. Let's put it that way. And so this is a simulation. It's a package that we call a lens that we developed at the Flatiron Institute, where I have kind of resources to do these kind of large-scale software projects, which is great. And what you're seeing here is for a type of motor that you do see in the spindle with kind of little microtubules that don't denucleate and go away. They're just kind of permanent, like in the experiments, the first experiments. You're seeing the formation of what are called asters, which are these little structures where you have the microtubules are polar. All the minus ends of the microtubules are being bound together by these motors, forming these kind of star-like, hence aster-like objects, and has a very beautiful kind of coarsening dynamics going in time. So, speaking of asters are very important for another problem that I have become very interested in, and this has to do with that first problem of transport and positioning of material inside of the cell. And this really started from work at NYU with Fabio Piano, who is a biologist in our biology department, and continued with a long collaboration with Dan Needleman from Harvard. And what you're seeing here are two asters that are moving nuclear material inside of the cell so it can assume what's called proper position, so that the nuclear material or chromosomes that it's carrying around can condense and then be divided by this mitotic spindle. That's really its job. I neglected to mention that. So that's been a provocation for developing of other methods, quite different methods, for looking at flexible objects, long, skinny, flexible objects, carrying payloads around inside of containers like a cell, let's put it that way, where you account for the full fluid mechanics of this and how it modulates all the forces that you have. And one of the questions that you try to answer with these types of simulations is questions of, if I have two different models, this is a contested question, if I have two different models of how motors exert forces on microtubules to move the genetic material around and position it, which one is correct? Which is the real one? Maybe it's both, maybe it's neither. At any rate, these two, you will see, end at the same place with proper position for the payload. That's rather discouraging. You didn't discriminate anything. But it turned out that these produce very different fluid flows on their way to positioning this object inside of this model cell. One model produces internal flows around the asters that come from the fact that motors are kind of internalized into the whole object and they're moving everything around. In another model, all the motors sit on the boundary and they produce flows that are basically just coming from the motion of this big array of things moving around inside and dragging the fluid with it. So, and then I teamed up with Dan again, and in his lab with a very gifted graduate student, Haiyan Wu, they reconstructed the fluid flows inside of these 50-micron little guys. And lo and behold, what we discovered is that the model with boundary-bound motors was the one that was really describing in detail what was going on in this problem of positioning things into the right place. Okay, now I'm already starting to hit a boundary, but I think I can finish this part. So, speaking of fluid flows, that's the way I do my transitions. Speaking of fluid flows, there's another beautiful flow problem in development, and this is in Drosophila oocytes. So these are pre-fertilized eggs, if you like. And while the egg is being developed in this egg chamber of the Drosophila, you see all these nurse cells over there on the left, they're the red-labeled fellows. This void, which is actually an active cell, develops, and at some point in that void, there spontaneously forms flows of cytoplasm, a big vortex moving things around, which we call a twister. These have been known for a long time, but it turns out that this twister is again associated with microtubules and motors and is a self-organized object, we believe, and I'll show you what I mean by that. So here's a basic physical model of a inside of this oocyte. It turns out that you have a bunch of microtubules that are bound onto the side of the cell, and there are motors that are carrying payloads up along these surface-bound microtubules, these kind of little elastica. So it turns out it's Newton's third law. You drag something up, you got to push something down. You push down hard enough, you can make all of these microtubules bend over like a big forest. They all bend the same way because they're all in the same fluid, and they all push on each other. And so we constructed a new mathematical model for that, which I'll just show you does what I want to do, but I won't say much more than that because I'm running out of time. But let me show you a simulation using these same types of computational fluid dynamics methods, and this is another package we developed called SkellySim, coincidentally. And this is a spherical cell where I have a couple thousand microtubules with a model for motor protein forcing. And let me show you what it does. Starts from an unstable place. They're all kind of pointing inwards. And this spontaneously forms a global state where all the fluid inside is just rotating around because these motors are pushing stuff around. There's a whole story about defects and things like this, which are all very fascinating to people like me. There you see the big vortex. What happens if you put it inside of a oocyte-shaped cell? That's a good question. They turn out to be very robust. There's a split view of one, and there it goes, forming one of these vortices again. Okay? There's a whole beautiful story that has to do with the subsequent motion of these twisters, which we're pursuing with our experimentalist friends. And I'll skip this part, except to just return to look at this problem again over very, very long times. That's the motion inside the oocyte of big, bands of microtubules waving around. It looks like seaweed in a tidal pool. It's really very beautiful. That's about on the order of an hour. And it brings me back to the applied math lab, where way back we looked at the flapping of flags, and that's an experiment that we did there, and we did theory for it. And the one thing I want to leave you with is there's this little lesson here. Those are two examples of kind of undulatory motion. And let me just say, sometimes the wind moves the flag, but sometimes it's the flag moving the wind, and that's what's going on in these oocytes. And some of you may know the story of Huineng, the Buddhist philosopher, settling an argument on flags between two young monks. If you don't, I ask you to look it up. And I'll stop there. Thank you very much.
Thank you. Questions? No questions. Okay.
Thank you. Thank you.
Thank you. Okay. Our second speaker is Arturo Casadevall. Oh, thank you. He represents Class 4 section on microbiology. He's from the Bloomberg. He's at Bloomberg Distinguished Professor, Alfred and Jill Sommer Professor and Chair of Molecular Microbiology and Immunology at Johns Hopkins Bloomberg School of Public Health. His title is Something New, Something Old, Antibody Therapy at a Time of COVID.
Thank you so much. Thank you for the honor of representing my division. I'm here for my primary section is 44, secondary section is 43. I figure I put them up because the microbiologists think I'm an immunologist, and the immunologists think I'm a microbiologist. And that's what happens when you have too many interests. Tell you a little bit about me. I have an accent. I came from Cuba, the middle of the island. My primary education was in Cuba. My parents took me out at the age of 11, and that was the best thing that happened in my life. I had the wonder to grow up in this wonderful country. We left by Spain. There you can see me on the right with a hand-me-down. Somebody donated some clothes, and I have a jacket on so I could look good. In those days, people dressed up to fly. Settled in Queens, New York, moved a lot, and came out of this with a great appreciation of history. So our theme of this talk is that history matters. What's missing in a lot of those steps is that the first 10 years in this country was a very disorganizing time. Just trying to figure out what I was doing. Certainly went to college at the only place we could afford. The city university was free then. My father didn't think I was going to amount to anything, so he sent me to get a degree in pest control operations. And you can see it up there right on my desk. I see it every day when I go in, and it reminds me exactly where I came from. He said he would invest $500 in me to buy 2 tanks of pest killers, insecticides. And that with that, I could basically do it and replenish the tanks. And in New York City, I would never starve, for there will always be roaches. But fortunately, in college, I discovered science. And this was, again, a wonderful time. I didn't come from a scientific family. And then I worked really hard to get good grades. And then my father insisted, I wanted to go to graduate school. My father insisted I had to go to medical school. But then, before I tell you that, I want to share with you 2 views of the history of science. The history of science bores most scientists stiff, but Darwin felt very differently, that it was essential for posing problems. And I will argue to you that the history of science is essential for understanding some problems and for avoiding foibles in pandemics. So the second biggest break, my dad wants me to go to medical school. I don't want to go to medical school. I want to is that I discovered MD PhD programs. And I applied, and one admitted me after 6 months waitlisted right before school ended. You know what happened there, somebody dropped out. But it was a huge break, so I trained in physical chemistry, and then I had my clinical training at Bellevue Hospital, and that was, a searing experience, because that happened right at the height of the AIDS pandemic. Mortality was close to 100%. Most people were dying a lot younger than I was. Nobody thought you would be able to make antiviral therapy, so what you wanted to do was to try to treat opportunistic infections. And I was very interested in doing something about this, so I basically decided I had to retrain myself to go into an immunology lab, and I wanted to develop antibody therapy. I knew that antibody therapies have been used at the beginning of the 20th century. They have been the first effective therapy. In fact, the first Nobel Prize was given for the treatment of diphtheria with serum therapy. So I didn't become a pest control operator, but I went after other pests, infectious disease. So I trained in infectious disease, and I went to the laboratory of Matthew Sharp. He's still active. He was Section 43. He gave me a project to work in immunoglobulin genes, but I wanted to try to do something useful to deal with this pandemic, and I was very affected by a patient that I took care of, that I gave him industrial doses of antifungal and still died an agonizing death with that fungal disease caused by Cryptococcus neoformans. But Matty was very concerned about me taking on this project, so he sent me to see his friends. So some of the advice are, if you want a career in academia, don't work on anything useful. Cryptococcus is a big pneumococcus. You're going to discover everything we already know. Work on relevant immunology, T-cell immunity, antibodies are done. But I had a bigger problem. And the bigger problem was that I was trying to make antibodies for organisms on the right column. And immunology told you can't do it. It doesn't work. And there's a quote from that time: antibodies in general are not a major fungal infection and protection against these fungal infections. Could this be true? So I went into the literature and tried to understand, how do people come to this view? And I realized that there was a logical error. And the logical error was that they had fell into the Aristotelian argument from ignorance. using the absence of evidence as evidence of its absence. Basically, because they could not show it didn't work, they concluded that it didn't work. So we took a new approach, monoclonal antibodies, make monoclonal antibodies to the fungus and immediately find out what the problem is. The problem is that you make good, bad, and indifferent, and when you put them all together, you cannot show the efficacy of the entire response. But that you can still make protective antibodies, and in fact, I took one of those into a clinical trial. Just to give you a sense, I spent about 10 years trying to sort all this out. The protective and the non-protective antibodies are very different. In this case, they basically lead to the deposit of complement, which is a part of the immune system, on the surface for the good ones and not for the other ones, and discovered things that were totally unexpected that you certainly couldn't have gotten from studying pneumococcus. Simply, antibodies were not done. Here, the antibodies bind to the capsule. They cause a conformational change in the capsule that allows the polysaccharide to bind directly to the complement receptor. So you have a brand new mechanism of phagocytosis. Now, we also took on the tuberculosis, mycobacterium tuberculosis. Didn't think antibodies work for it. Make the first protective antibody working with Barry Bloom. Know that it binds to Rabibanam, and then made a vaccine. So the bottom line is you could protect by either mechanism. Immunity was whole. There was no need to divide it. And then when you look at history, you realize that immunity had been whole in the past. And then it was divided by reductionism. And then as you began to put all this stuff together, it's very clear that you could protect by many mechanisms. There isn't necessarily a duality in any of this. So today, this is all those controversies is gone. And I'm going to share with you two quotes that as I get older are increasingly disturbing. So one of them is, a new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because the opponents eventually die. And a new generation grows up that is familiar with it. And the next, even more disturbing, is from this one, Paul Samuelson, science makes progress funeral to funeral. So that may be true, but the sad thing is that in every generation, scientists don't take care of their history, and history is lost. And the problems are discarded. Science is lucky. It can work in the absence of history. You cannot practice law without knowing history, but you can do cutting-edge science just with what you know today. But I would argue that history allows you to phrase problems in a very different way. So let's go to coronavirus 2018. So it's January 2020. I'm an infectious disease doc. I know this is not controllable. I'm reading the papers, and people are talking about vaccines, antivirals, but nobody's talking about plasma therapy from the beginning of the 20th century. How do you get the word out? Well, I'm an editor of a journal. I can certainly put it in a journal, but no, you want to get it into the general public. So I write an op-ed. I send it to New York Times. I send it to Washington Post. Nobody wants it. But like everything in life, everything is luck. So I sent it to the Wall Street Journal on the day that the market tanked. And this was good news, so they published it. What did I write about? I wrote about, I pulled out a case from history. In 1935, this individual, Roswell Gallagher, who in the 20th century would go on to be a major person in adolescent medicine, stops an outbreak in that school that is still in Pennsylvania by taking the serum from a boy who's recovered and treating 60 others that have been exposed. So we also know that this had been used in 1918 with success. And in March 2020, things moved very fast. So then I had this op-ed, and I sent it to all my friends, including some of you in the audience. At Hopkins, we organize very rapidly, and we try to set up clinical trials. And we create then a convalescent plasma project. We get all our friends together and begin working to do this. And within 3 weeks, the FDA allowed compassionate use, and within a month, the first patient had been treated. What had happened during that time is I had my 15 minutes of fame. And the more I went on television and the more I gave interviews, the more people asked the FDA for permission, and the FDA decided to end up creating what I call a big registry in the country that was headed by Michael Joyner at Mayo, and this will become a really, really important source of early information. Convalescent plasma in the United States, in fact, has the echoes of what is known as Tocquevillian civic association. If you're worried about the problems in the United States today, I urge you to read this book. In 1831, the echoes of everything that's happening was happening back then. in different ways. But what he noticed was that whereas in Europe, everything happened from the top down. The king sent a representative to the town to fix the water well. In the United States, as civic associations took care of things. And this is a fundamental strength that we have in this country. So what happened here is people just organized. Blood bankers, the FDA provided guidance, groups began to recruit donors, and within a couple of months, 90,000 patients have been treated. And the United States went from 0 units to using 20,000 in about 5 months. The United States does it different than the rest of the world. The FDA allows its use based on history. The rest of the world says, no, you must do randomized controlled trials before we do it. This is going to cause tremendous problems. In the United States, the use increases rapidly, but criticism of the FDA rises. How could you allow this to be done without evidence of safety, evidence of efficacy? The rest of the world sets up randomized controlled trials, but sets them up without knowledge of history. So what they do is they treat patients that are too sick. Antibody will be shown not to work in those situations. So in the United States, plasma takes off following the number of cases. And by the spring, we were able to show that plasma was safe. There have been all these concerns, immunological concerns about antibody-dependent enhancement, triggering cytokine storms, type 3 sensitivity, giving antibody to somebody who has antigen. None of that turns out to be the case. Plasma is as safe as regular plasma. What about efficacy? Can you get data and efficacy from a registry? So, working with the FDA, the first 35,000 patients get analyzed, and you see a very strong signal emerges. Those who are treated early are less likely to die than those that are treated late. But more important from a scientific point of view, you see a dose response, and you know that a dose response is a very strong evidence for causality. The people who receive high blood plasma were less likely to die than those that receive low-titer plasma. And this was subsequently published in the New England Journal of Medicine. Unfortunately, this is what happened. Everything became politicized in the late summer of 2020. We're not going to go there to say that it's not a good thing when science becomes politicized. If you look at the use of plasma in the United States, the more plasma the United States used, the fewer people died. And you can take that data and you can calculate, by the end of the first year, more than 500,000 people have been treated. You can calculate what would have happened had the FDA not allowed it to be used. You can estimate another 100,000 deaths. But here comes the clinical trials from all over the world, and that's the big red cloud up there. Red means it doesn't work. Yellow means it's some evidence of efficacy. Green means it worked. And what you can see is that those trials that were set up treating early and people who weren't very sick, you get evidence of efficacy. If you wait till it's late, it doesn't work. There is no plausible mechanism by which antibody is going to reverse lung inflammation. There is one red at the bottom, and that's an outlier. That trial was done in Spain using methylene blue treated plasma. And there is data from the 1930s that methylene blue generates free radicals that can damage antibodies. So what was forgotten was the history lesson. This is from 1938, pneumococcus, 1946, diphtheria, 1913, meningococcal meningitis. You have to treat early. You have to treat in the first few hours, first few days. And this is a statement from that time. It is a fundamental principle of all serum therapy that to obtain the best results. This serum must be given early in disease. This was validated in COVID. This is a study in Texas. You could see, look at the two curves. The curves at the bottom is after 3 days. They're super pretty, they don't separate, but the ones at the top separate. And in fact, you can identify a 44-hour window by which you must treat your hospitalized patients. This is the most influential trial done in Europe. 50% of the patients are randomized, and after they get randomized, they get treated before 48 hours, 50% after 48. In fact, 50% had their own antibody. So then this gets published, and this hit the American physicians. Plasma doesn't work. And this is what happens. Plasma use drops dramatically at the beginning of 2021. It drops at a time in which we don't have any other option. The sad thing is the American doctors were using it right. They were treating people on admission. And from this, you can estimate that about 30,000 excess deaths in the winter of 2020, 2021, as a therapy is withdrawn without having an alternative. At Hopkins, we set up the trials as an outpatient. This is the credit to David Sullivan. It's a large, randomized, double-blinded, all the things that everybody wants to see in evidence-based medicine, and the result is, as expected, it works. It had a 54% reduction in mortality, and 2 was published. Today, convalescent plasma remains under emergency use authorization in the United States with the lowest of all monoclonals. It is the only antibody therapy available, and it's fine use in the immunocompromised. Back to the bench, I have one slide after this, just to tell you that new basic science can even come out of this effort. It turns out that people think that the way that the antibody neutralizes the virus by interfering with its binding. Well, this is a paper that will hopefully be accepted very shortly, in which it shows that the antibodies are working as catalysts too, and they're chewing up the virion and destroying. So about 50% of the activity in plasma is coming from antibody-mediated catalysis. So some final thoughts. What is old is new, and what is new is old. If the current bird flu hits us, you can be sure that convalescent plasma will be rolled out again. But a good legacy of COVID is that we know how to use it. The history of medicine and science can help frame questions, and I thank you for electing me to the National Academy of Sciences.
Do you have any questions? Come on, I told all these people that the sessions generate these wonderful questions. Guess we're tired today. Okay. Well, thank you so much.
Thank you.
Our next speaker is Gunnar. She's a Regents professor and distinguished McKnight professor of developmental psychology, Institute of Child Development at the University of Minnesota. She represents Class 5 section on psychological and cognitive sciences, and the title of her talk is The Stress Buffering Power of Parents Until Adolescence. I think we all know that.
Thank you. I want to thank the Academy, of course, for inducting me into its membership, and my class for selecting me to share some of my research with you today. I'm a developmental psychobiologist. I study the physiology of stress, its regulation, and how it impacts brain and behavioral development in children and adolescents. Now, much of my research has to do with this hormone, cortisol, produced by the hypothalamic pituitary adrenal cortical system, HPA for short. It's a powerful steroid hormone that is essential for everyday normal functioning, but critically important when we must meet the challenges of threats to our physical or our psychological self. It's a steroid hormone, and so like other steroids, it is lipid soluble, and that means it gets into all the cells of our body where it regulates hundreds and hundreds of genes that are responsive to it. But as a lipid-soluble steroid, it also gets into our saliva, which is why I can collect spit to assay how much cortisol children's bodies are producing, and which is why I've been known as the spit lady for many, many years. Okay, the Academy has asked us to share some of our story about how we got to the research we do, and I think that's important especially for young people who think that we all knew what we were doing from the beginning, which we all know is not true. In fact, I started college as a music major. I went through many iterations of what I might to do until I discovered research on hormone behavior relations, and I was hooked. But at that time, I was not interested in cortisol and stress. I was interested in this hormone, testosterone. And what I wanted to understand was how variation in this hormone during fetal development might be related to the development of sex differences in behavior as we grow up. And to study that, I went to Stanford University, and I worked with Eleanor Maccabee, a National Academy member, who at that time was collecting umbilical cord blood from babies, at birth, of course, and having it assayed for testosterone and other sex steroids. And then she was following the babies as they developed. And when I arrived at Stanford, the babies were turning a year of age, and Eleanor tasked me with looking at sex differences in fearfulness. In essence, she wanted me to scare babies. And the way I was supposed to do this was to sit them in front of this toy monkey, turn the monkey on, it would clap its cymbals, and I would record whether the babies cried and went to their parents, which most of them did. And that confused me, because I knew year-old babies. I knew that if you gave them something to bang with, they would make way more noise than that toy monkey made. So what was so scary? And then I thought, well, you know, they're not the ones turning it on. So I ran a study where I randomly assigned babies and trained them up so they could turn the monkey on themselves, and I had another group that were a yoked comparison group, and sure enough, the babies who could turn it on themselves thought it was hysterically funny, and the ones who didn't were frightened. And oh my gosh, I was going to get myself a PhD. Because at that time in developmental psychology, no one was talking about fear and its development in relationship to control in human children. And then I started reading in the literature on the adult literature and the animal literature, and of course I had rediscovered the wheel, because in those literatures it was well known that fear and control were tightly linked, and more importantly, for me, it was well known that control had a lot to do with the regulation of stress and activity of that HPA axis with the production of glucocorticoids. Furthermore, I discovered that just 2 blocks from where I was in the psychology department, Seymour Levine over in the medical school was showing that the experiences that rats had early in life was shaping the development of the system and influencing how fearful they would be throughout their lives. So I finished my PhD studying control and predictability, its close cousin, and fear in human children. And I went over to the medical school and did a postdoc with Seymour Levine studying rats and monkeys. Now, no offense to my rat and monkey friends, but I did not want to spend the rest of my career studying rats and monkeys. But in 1978, when this was, we knew that the assays for cortisol that you would need to do in plasma and urine at that time were being adapted so that we could collect saliva and assay it in saliva. And I knew then that I would be able to take and discover really what you might think of as the human chapter of our mammalian story about early life stress and development, and do it with kids. Now, when I started my research with children, we knew next to nothing really about how this system worked in human children. We know a lot now. There are a bunch of research stories I could tell you, but I think the most important one to share is the story about how the formation of attachment relationships between parents and children, or parenting figures and children during development, provides this powerful buffer of the stress system during the period of time when children's brains are developing rapidly, and in a sense is protecting our developing brains from the overproduction of this hormone. So that's the story you're going to hear. Now, one of the first things that we learned was that, and of course, we can do it in spit. One of the first things we learned was that newborns have incredibly reactive HPA systems. You do anything to a newborn that causes it to cry, and it elevates cortisol. But that's okay, and it doesn't last very long. In fact, at around 3-4 months of age, you see a marked tightening of the system, which we now think is due to maturation within the HPA axis itself. And you can see that in this study that we did, where children, we were following them during their well baby exams with the shots that they get afterwards. And as you can see, at 2 months, there was a really big response, then a more tightly regulated response at 4 and 6 months. And then this really amazing, weird thing happens, in a sense. And we know it happens between 6 and 12 months because of additional work we've done. And that is that even though the baby is going to the doctor, getting a shot, crying and clinging to its parent, the HPA axis has ceased to respond. And the timing of that was really suspicious, because between 6 and 12 months is when babies begin to organize their security-seeking behavior around one or a few people. And this is called the formation of specific attachments. And especially if those relationships are secure, if the baby's securely attached to that adult, the baby now has a powerful way of predicting that when it signals that I am distressed and frightened, I'll be safe, I'll be taken care of. And we began to wonder whether Perhaps the formation of the specific attachment, especially if secure, was allowing this powerful regulation of the axis. Now, most babies form secure attachment relationships with their parents because most parents are responsive to babies' needs when they're scared and frightened. But some babies don't, and Mary Ainsworth developed a situation called the strange situation that we can use to assess the security of attachment. It involves a lot of strange comings and goings, and we record how well the baby, or how poorly the baby, is able to use the parent as a secure base in that situation. And we've done this in a number of studies, and then we followed the children in other situations, either in our laboratory or in the real world, where they experience a stressor or a potential stressor, and we observe whether the presence of that parent is able, in the secure relationships, to buffer that system, and indeed, the parent is powerfully able to do so. And here's just an example of one of our studies where we literally sent in the clowns. These are 18-month-olds. Never hire a clown for your toddler's birthday party because some kids are totally terrified of clowns. As you can see on the right-hand side of that graph, those who were frightened but were securely attached to the parent who was there with them showed no elevation in cortisol, no more than the kids who thought the clown was funny. But if they were insecurely attached to the parent who was with them, it's like they had a leaky buffer. They were producing elevations. Now, one of the things we have wondered is how long does this last in childhood? And to do that, we had to move from sending in the clowns to the most commonly used stressor task in stress research, which is giving a public presentation. And I'm handing out cotton rolls to collect saliva from all my colleagues over here. This task, the child or adult stands in front of two judges, gives a speech while being filmed, and told that film is going to be shown to kids of their own age who will also judge their speech. It elevates cortisol, okay? To see whether the parent could help buffer that, we merely had the parent help the children prepare for the speech. And then we sent them to the waiting room, and the kids finished the task on their own. And compared to preparing with a nice experimenter, children who prepared with their parent all the way through middle childhood showed very little response to this public speaking task. Parent power is huge. And then you get to adolescence. And in adolescence, no longer. No effect of preparing with your parent. And we now know that that's occurring with puberty, about the midpoint in puberty, about stage 3. Suddenly, it's like the parent gets booted out of the kid's HPA system, is no longer a part of that, and children need to go on and regulate and learn how to regulate the system on their own, or they'll never leave home. That timing is also suspicious because it's about that point, that midpoint in puberty, where we see this big uptick in children developing depressive disorders, which we know are also related to dysregulation or problems regulating this system. But that's a story for another day. I'm going to go on and take a look at what happens here if that buffer is not just leaky, but it's non-existent. For the past 20-so years, my students and I have been studying children who lack an attachment buffer because they start their lives, their first few years of life, in orphanages or other institutions around the world before they become adopted or fostered into families. Now, the United States went through this remarkable period of about 10 years when we adopted over 300,000 children from orphanages around the world. That's stopped now for two reasons. One, we've tightened up the regulations because we know that not all of those children were orphans. Some had parents who were looking for them. And we've come to understand the challenges faced by children who have been adopted transracially. But nonetheless, there are a lot of individuals in this country who started their lives in orphanages, and there are thousands and thousands more around the world still who are growing up in orphanage-like institutions. And in an institution, it is very hard to provide for babies, brains and bodies, what they need to develop. Longer you're in an institution, the more you lag behind in your physical growth, and in your behavioral development. And we know from studying kids in orphanages that your HPA axis also undergoes this a change in its regulation. Specifically, early in the morning when cortisol levels should be high to help you prepare for the day, they're low. And late in the evening when they should be almost non-existent, they're higher, which we know disrupts circadian rhythms and sleep. Okay, once children are adopted or fostered into families, they show remarkable rebounds in all aspects of development. But the effects depend in part on the degree and duration of deprivation, and with longer degrees and more deprivation, that signature lasts longer. And we certainly know that this is true for our HPA system. So children adopted beyond 18 to 24 months, which seems to be a sensitive or a critical period, continue to show that hypofunctioning, hypoactivity, which may have been nature's way of trying to protect that developing brain without an attachment figure present, but it comes at a cost. Now, what I mean is we see lower than typical levels early in the morning, and when we stress children or attempt to stress them, they don't show as much of a response. Okay? So, this coming at a cost, we have found that these children who are showing this hypofunctioning are more likely to develop disordered attachments, more likely to develop problems with attention and behavior regulation, and through these pathways, more likely to have problems with teachers and peers. But I've already told you that puberty produces big changes in the social regulation of the axis. It produces a lot more than that. There's a load of physical growth, as you know. There's a load of brain development. And during periods of time when our bodies and our brains are developing rapidly, they're also more open for being influenced by what's happening to us. And this may be true for the stress system as well, because there's animal research that shows us that if you stress a pubertal rat, the effects are bigger and stronger than if you do the same stressor to an adult rat. But periods of plasticity cut both ways. So if negative things can have bigger effects, positive things might have bigger effects also. And we began to wonder whether for these kids with these hypofunctioning HPA axes years after adoption, as they started going through puberty, perhaps that system, the body would open up, sample the environment again, recognize that, boy, I'm in a more benign situation. I can afford to mount this stress response if it's needed. It burns calories and does all these things. So perhaps with puberty, they'd recalibrate. Now, to answer that, we again went back to our public speaking task, but this time without parents helping to provide that buffer. And we had nurses examine the children to determine what stage of puberty they were in. And we tracked them over a number of years as they went through puberty. What you see on the right side of this graph is these are the non-adopted kids born and raised in their families in Minnesota. We are above average. They don't show any change in response as they go through puberty. The story is on the left side of that graph. These are the children adopted from orphanages beyond 18 to 24 months. And as you can see on that bottom line, these are the kids who have not started into puberty yet. They have low levels that just collapse during the stressor test. They report being very scared. As they go through puberty, the system begins to open up and is responding more and more until it's responding just like kids who have never experienced deprivation. There's this amazing recalibration. Now, this is likely a good story for some things, but it's also a big challenge for these kids because, in essence, they have gone from driving a Volkswagen stress system to driving a Ferrari stress system. And their brains are not used to seeing this increase in cortisol when things are threatening. We expect that that would mean that perhaps the amygdala, important in fear and its regulation, might be being more reactive. In fact, what we're seeing is children who recalibrate more are showing more anxious symptoms, and we've followed them into late adolescence and early adult, and that is true. The bottom line for all of this is human development begins with supportive care for children from birth and for parents and mothers before that. Now, this is not just a story for us to tell each other in the academy. It's important to get this information out. And I've been involved throughout my career in trying to translate work for practitioners and policymakers, which is a big task of the academy as well. And I look forward to continue my work in science translation as a member with you all. Thank you.
Questions? Okay. Can't tell if somebody's...
No, people are leaving. There's no question.
No?
Is there a question?
Okay, we have a question.
Oh, Rochelle.
Yes.
Are your conclusions independent of population or countries?
Yep. We've seen it, little girls from China, kids from Russia. Yes, they are. Now, yes, they are, at least in terms of the HPA axis.
Right. Thank you. Our final speaker for today is Yvette Perfecto. She is the James E. Crowfoot, I think it's Crowfoot, Collegiate Professor of Environmental Justice, the School of Environment and Sustainability, University of Michigan. She is a representative of Class 6, section on environmental science and ecology, and the title of her talk is Biodiversity and Autonomous Pest Control in Agroecosystems.
Thank you very much. It's an honor for me to be here today, and especially representing my class in section 63, which is environmental science and ecology. So I am an agroecologist. I am an ecologist, but specifically within ecology, I am an agroecologist. So you might ask, what is an agroecologist? Well, if an ecologist is something like a detective of nature, an agroecologist is a detective of nature that works in agricultural systems. As an agroecologist, I try to solve the mysteries of the natural world and apply what I learned from doing that research to basically enhance agriculture, to make agriculture more sustainable. And here I am doing some of my agroecological detective work with my PhD student, Iris Rivera. To become an agroecologist, I went from Puerto Rico to the University of Michigan. I got a master's degree and then a PhD. But some of the best agroecologists that I have met in my life are farmers. Now, this is the University of Michigan School for Environment and Sustainability. As I mentioned before, some of the best agroecologists that I met are farmers, and some of them with very little education, with very little formal education. Farmers, by the very nature of what they do, are constantly trying to understand nature. And they use that knowledge that they develop to manage their farms. Traditional farmers have the additional advantage of generations of accumulated knowledge based on trial and error and experimentations on farms. One thing that stands out in my mind when I talk with farmers from different parts of the world about their farms and what they do is the importance they place on biodiversity. Whether organic farmers from Michigan or traditional farmers from Chiapas, they all talk about how biodiversity is working for them. Now, in trying to understand this phrase, working for them, basically, sometimes it's pretty obvious what they mean. But, so for example, it could be that they see a diversity of pollinators and they know that a diversity of pollinators is good for pollinating a diversity of crops. Polycultures, that is growing more fields, multiple fields multiple crops in the same field at the same time, yield more than monocultures when they have different nutrient requirements. Genetic diversity can keep diseases, this is polycultures here, and genetic diseases can keep, genetic diversity can keep diseases from spreading. So these are things that the farmers know, no? The idea that biodiversity works for farmers crystallized in my mind when I was working in coffee plantations in Mexico. One day I was walking with Don Walter, who you can see here, and I noticed this outbreak of caterpillars. And I mentioned it to him, and he looked at me and smiled, and he said, well, Yvette, don't worry about that because the migratory birds are starting to arrive and they will take care of that. And sure enough, in a couple of days, the migratory birds were feasting on these caterpillars and ate every single one of them. Don Walter's observation inspired us to experiment on this, do a big experiment to study the effect of birds on insect pests in coffee. The experiment consisted of putting this netting over coffee plants and then sampling the insects inside and outside and sampling the damage of the coffee plants inside and outside the nets. We did this in collaboration with the late Russell Greenberg from the Smithsonian Migratory Bird Center. What we found surprised us. First of all, not only birds, but also bats were eating the pests in coffee. And second, one other thing that we discovered in this experiment was that birds and bats were having not only a direct effect by eating the pest, but also an indirect effect through their effect on the spiders that eat the parasitic wasps that eat the pest. No? We learned two lessons from this experience. First, that nature is more complex than we think it is sometimes. And second, that farmers and scientists make a great team. Farmers and scientists have very different type of knowledge. Farmers have an intimate and deep knowledge of their environment, while scientists have a broader, perhaps universal type of knowledge. And it is the combination of these two types of knowledge that I think is extremely powerful, and in my opinion should be the basis of the new agriculture. As I mentioned before, when farmers are talking about biodiversity working for them, sometimes it's pretty obvious what they mean, but other times, basically they don't know exactly how biodiversity is working for them. And it is at those times when having an agroecologist in your farm becomes handy. Let me take you then through a journey that we have taken in the discovery of how exactly biodiversity is working for the farmers. No? Some part of our agroecological detective work in the coffee farms in Mexico. So this is the Azteca ant, and the Azteca ant nest on the trees, on the shade trees that are grown with the coffee, and they forage on the coffee plants that are surrounding, that are close by the tree. They tend these tiny insects that are called scale insects, and as you can see here, those are the scale insects. They tend those scale insects on the coffee plants, and they produce a sugarly solution called honeydew. So it's like a honey-type solution, and they excrete that honey-type solution. You can see it here. They're excreting the honey there, and here you can see the ant taking that honey. And in exchange for that, the ants protect the scales from their natural enemies. And one of their natural enemies is this lady beetle. Now, this lady beetle is a voracious predator on the scale insects. And here you can see the larvae of the lady beetle, and they have all these waxy filaments that you can see there, and those waxy filaments protect them against the action of the ants. And here you can see two ants trying to kill the scales, trying to kill the lady beetle larvae, but they're not successful because of all those waxy filaments. And so what happens then is that because they're protected from the ants, they can live in these patches that have a lot of scales. And since the larvae is protected, they can prosper in these patches. No? The adult beetle, on the other hand, is constantly harassed by the ants and sometimes killed by the ants. In this video here, you can see the ants tending the scale insects. And now, from what I told you before, we know that it is good to have lots of beetles on your farm because they are predators of the scale insects. We also know that the plants with ants are really good for the development of the beetle. And finally, we also know that the adult beetles cannot get close to the plants that have ants because they get harassed and killed by the ants. So this brings up two questions. First, how can the adult beetles prevent their eggs from being eaten by the ants? And second, how can the adult beetles lay their eggs on plants that have ants? Because the ants are constantly harassing them. So, as good agroecologists, we set out to solve this mystery. We found the answer to the first question one time when we were examining scales under the microscope, and we lifted one of the scales and saw this. These are beetle eggs that are hidden under the scale. So this is a very smart mother, no? That hid the eggs under the scale, and so the scales are protected, the beetles are protected, and once they hatch, the larvae start eating the scales right away. The second question turned out to be even more fascinating. Now, it turns out that there is a fly that parasitizes the ants. This fly that you can see here. The fly attacks an individual ant and lays an egg on the head of the ant, and then the larvae gets inside the head of the ant. It develops there. Eventually, the ant's head falls off, and the little fly emerges through the mouth of the ant. That's why this fly is called a decapitating fly. And it must be a terrible way to go for an ant. It also happens that the fly has really bad eyesight and needs the movement of the ant to be able to locate the ant and parasitize it. Therefore, as a protective measure, when the fly arrives, that you can see here, then the ants become catatonic and paralyzed, and release a pheromone that tells the other ants to get paralyzed as well, and the activity of the ants goes down. And so here you can see the posture that this is a cartoon of the posture that the ants take when they are attacked, when they get paralyzed, basically. Now, here's an illustration of what happens. A fly attacks an ant, it becomes catatonic, releases a chemical that tells the other ants, the nestmates, that are in the vicinity, to get catatonic as well and get paralyzed. Now, guess what? The gravid female beetles have been able to decode the chemical language of the ants. And they effectively eavesdrop on the ants' conversation, thus finding out when the flies are going to arrive, when the flies arrive, and use that as a window of opportunity to hide their eggs under the scales. They have time, and there are not many ants around because they are catatonic, and they hide their eggs under the scales during that window of opportunity. Isn't that amazing? It's truly amazing. So putting all these pieces together, these pieces of the puzzle together, reveal a complex system of what we call autonomous pest control. The ants obtain energy from the scales. The beetle, both larvae and adult, eat the scales. But then we have a higher order effect because the ants disrupt the ability of the beetles to eat the scales and to oviposit. However, there are flies that attack the ants, that parasitize the ants, but more importantly, they interfere with the ability of the ants to interfere with the ability of the beetles to eat the scales and oviposit. This is a second level higher order effect. And these cascades of interactions in the patches where the ants are basically result in the biological control of pests in the entire farm. We call this autonomous pest control because the control emerges from the complex interactions that are embedded within the biodiversity of the farm. The system also has a spatial component. Only about 5 to 10% of the trees have nests of Azteca. However, the ant patches are the refuge of the beetle, which is the main biological control for the entire farm. But when the farmers encounter a patch that have the ants, guess what? They basically, when they encounter a patch, they basically want to get rid of the ants because the ants are tending the scales and they can see that. And the idea is that the friend of my enemy is my enemy, no? So they try to kill also the ants and get rid of the ants. So how do we communicate these complex interactions to the farmers and convince them that they shouldn't eliminate the ants, that the ants have a positive effect on the farm? Well, human beings have been playing games for thousands of years, and we figure, okay, well, maybe it would be a good idea to have the Azteca system in a game. And so we developed, with our colleague Luis Garcia Barrios from Ecosur in Mexico, we developed the Azteca chess game. And you can play this game if you were interested, let me know. It is a board game for understanding the complex ecological interactions in the coffee system. And we have done workshops with farmers in Puerto Rico and in Mexico where farmers play the game and discuss the complex interactions that are going on. Now, thanks to one of the farmers that is also happened to be also a wood carver, we have the deluxe version of the Azteca chess. He, on his own, came up with this idea and carved this beautiful game. Now, let's go back to our coffee system. So coffee can be produced in all these different ways, from the very diverse system that you see on the top to the coffee monoculture, and biodiversity decline along that gradient of intensification. In these very intensified systems, monocultures, the Azteca ants disappear because the Azteca ant is an arboreal ant and it needs the trees to nest. So the ant disappear, and we have done, our labs have done research that show that the Azteca is implicated, the ant is implicated in the control either directly or indirectly of these 4 main pests that you see here. And I don't know where the pointer is because I haven't been pointing. Oh, here. So these four pests. And so when you eliminate the ant, you're also basically you could stimulate an outbreak of the pest. Now, so when farmers ask me what control pests in the coffee agroecosystem, I tell them complex networks of ecological interactions. And yes, indeed, biodiversity is working for you. So for agriculture to be based on knowledge does not mean that every farmer needs to understand all the intricate, complex interactions going on within his or her farm. What farmers need to know is that biodiversity can help prevent pest outbreaks. The details can be extremely complex, but the overriding principle is simple. And the reality is that many farmers, as I mentioned at the beginning of this talk, many farmers appreciate biodiversity in general and the complexities embedded in it and are moving toward more agroecological diverse systems. Among the main players of these initiatives is the global peasant movement called La Via Campesina, or the Peasant Way, which has as its main goal, or one of its key goals, a more ecological approach to agriculture. This organization represents millions of small-scale farmers the world over. And as such, it may very well catalyze a turning point for agriculture, a turning point toward a future in which scientific ecological knowledge in combination with local traditional knowledge becomes the foundation for agriculture. A new agriculture that no longer is intensive in agrochemicals and in fossil fuels, but rather intensive in thought, knowledge, and wisdom. And I'm looking forward to that future. And with that, I want to thank the many, many, many graduate students and undergraduate students that have worked with us for many years. And in particular, I want to thank my partner, colleague, partner, friend, and husband, John Vandermeer, who is an essential part of this research as well. And his lab and my lab have been doing this research together. Thank you.
Do we have any questions? Okay. That's it. Well, thank you all, and let's thank all our speakers.
He has a question.
Wait, hang. You have a question. Go ahead. Come on, Jenny Lee. We have one question, at least.
For farms that lose the beetles or any part of their system, is there a way to bring the insects back?
I'm sorry, I didn't hear.
So if a farmer, you said that they try to extinguish the ants, or if they try to extinguish the beetles, is there a way to bring those insects back to like rebuild the system?
Yeah, you can definitely breed the insects, the beetles, and introduce them. But without the ants, the beetle is not going to be able to survive, because one thing that I didn't mention is that the beetles are attacked, the larvae is attacked by a parasitoid, and the ants inadvertently protect them against those parasitoids, because they're tending the scales and they scare away any parasitoid that come close. And since the beetle larvae is with the scales and the ants protect them against the parasitoids. So if the ants are not there, the beetle is not going to be able to prosper. So you have to have the ant for the beetles to be able to prosper in the ecosystem.
Okay, another question. All right.
Very much, Yvette. Have you ever had the opportunity to take a monoculture, the worst of your biodiverse gradient, and then reclaim it with natural colonization or with implanting? And if so, how long does it take to restore a fairly biodiverse system?
Yeah, great question. And there are studies that we haven't done those studies, but there are studies that have looked at reforestation of coffee monocultures. And we have looked actually at bird populations and how the birds recover after. And it takes several years for the birds to start coming back, the different species that are lost during that time when there's a monoculture. But the entire ecosystem might take much longer, because there are many different species that are interrelated, and we still don't understand all the relationships. Part of what we do in our lab is trying to understand all these different relationships that are going on in very diverse farms. So when you eliminate a lot of that biodiversity, it will take a very long time for those interactions for those species to come back, and then for those interactions to get reestablished.
One more question.
Is there a plant genetic component to this? Does this system operate also in Robusta coffee as well as in, so what part does the plant play in a sense, selecting the system, if it does that at all?
Yeah, that's a really interesting question, because this system, as you know, it's very complex. It looks like, it took millions of years for it to evolve. However, coffee is an introduced plant that has a history of about 200 years in Mexico. And so I think that the system evolved in the natural system, and then when coffee became prevalent in that region, they started colonizing, the different organisms started colonizing the coffee. Coffee is affected by these scales insects or other scales insects, no matter what variety it is, or species. Robusta coffee as well as Arabica coffee has the same scales. Yeah.
Okay. So if we can thank Yvette and the rest of our speakers from today.
Thank you.